Hello, everyone. Today, I'd like to introduce Dr. Geetanjali Bendale. She is a research scientist working on nerve repair at the Virginia Commonwealth University and also has a PhD in bioengineering from the University of Texas at Dallas. Can you tell us your story? I'd love to hear a little bit more about your background and your journey and what brought you to where you are today.
Almost over a decade ago, I came to the US in about 2012. I have a background in engineering. It was biotechnology, so a mix of biomedical engineering, instrumentation. My undergraduate degree is from India. So I wanted to pursue a master's degree. I was always interested in research, so I wanted to do something in the realm of research bioengineering. That's all I knew really about bioengineering when I decided to come to the States. So I came in to the University of Texas at Arlington to do my master's in bioengineering. I got my master's, and during my master's, really, I got introduced to research, like the real research, lab-based research that is, much more elaborate than I knew about. I got intrigued about what research actually is, what are the ways you can really contribute to scientific research. And that led me to my future PhD lab. At the moment, I was a graduate student, a master's student working with other PhD students and helping them in their projects in the lab. And that really gave me a full experience of what research is and would be like as a PhD student and what that would lead to. So that is really the idea I had about doing a PhD. And so that then led me to my lab at UT Arlington first, and then my PI moved from there to UT Dallas, which is really just 30 minutes away. But that's where I actually started my PhD program at UT Dallas in the biomedical engineering department. And at the time, the biomedical engineering department was fairly new, not that new, but we moved into this really beautiful new engineering research building. I worked on a range of different projects during my graduate work, and the kind of good and bad of not having a fully funded study really led me through twists and turns of different projects throughout the 5 years of my PhD, which made me really, the kind of scientist I am now, which back then did not seem very, very nice, because I wanted to have, a specific thesis, a specific project. And so I worked on multiple things in nerve repair regeneration, but also on the bioelectronic medicine side, which was kind of picking up at the time, developing new electrodes, testing these new electrodes, how to implant these new electrodes. So that's something I did on the side, trying to figure out what I was going to do. So I worked in both. My thesis ended up being in peripheral nerve regeneration. And so that's something that I worked on. I looked at different growth factors that helped with better regeneration. That's what my thesis was on. And, like every graduate student does, I did a lot of networking when I went to conferences. And through one of the people who I met at a conference, I was referred to for this job that I have now. And so I talked to them. They said they were looking for someone who had some experience in nerve repair research. And it kind of really came together very well about 5 years ago now when I graduated. And this is the first job I took out of grad school, and this is where I am right now. So I'm, very happy where that path took me. It was a pretty conventional path in the sense that I did my bachelor's, I did my master's, I did my PhD, and now I'm in an academic setting. But it was unconventional in the sort that I did not have a specific project when I started my thesis, a fully funded specific project. And so I had to kind of carve my way out trying to make a project for myself. But what that gave me was experience on multiple different projects that kind of connected me to both, the basic biology, neurobiology of nerve and regeneration, but also the bioelectronic side of what happens when you modulate a nerve. What I needed to know about the nerve really gave me the basis of understanding what happens when you want to change how the nerve functions. So I'm glad that I had to go through that. It was often as a graduate student very stressful, but I'm thankful for that experience now that, it is useful for me when I try to troubleshoot a lot of the things in the lab that I, work now. So I now am a research scientist, a full-time research faculty in the orthopedic surgery department at Virginia Commonwealth University. I work work with the surgeon scientists who are, few to be found, but very, very important when you do this kind of research is to have this clinical perspective. And so I think we make a really good partnership work because I do a lot of the basic science work. He puts in his clinical perspective and how better we can shape our basic science research so that it's translational. So it's more translational. We're always thinking about how this goes into patients. And so where I am right now, I'm working on very translational ideas. So trying to solve the basic science questions so that we can make it more translational. So I think I'm in the sweet spot of translating technology into the clinic, and I'm happy to be doing that work at the moment.
Yeah, it sounds like it's been a really exciting journey so far, and I can totally understand how that would be useful to have that background experience that you mentioned, being exposed to many different kinds of projects during your grad school time. It definitely seems like it was a good thing, like when your PI moved, that it was pretty close by, within driving distance. So that way you kind of still knew the neighborhood and knew the town and everything. That's exciting to be involved in kind of the translational side of research that you're working on now, and definitely super useful to be collaborating with these surgeon scientists, as you mentioned, to be able to see like the clinical patient side of things like more closely and have really good access to that. So yeah, definitely excited to hear about the current things you're working on. But before we dive into that, I was wondering if you could take us through what led to your initial interest in neuroscience and neurobiology, and also like the nerve repair side of things. Like, was this something that you were, always interested in growing up, or did you discover it during undergrad or during your graduate school?
Okay, so this kind of leads to a little bit of a backstory. So the reason I got into a bachelor's of, bioengineering, biotechnology. So back then, where I was in India, we didn't have a bioengineering program. It was called biotechnology because it mixed bioengineering, but also some food technology. So it was a new program. So that's where the reason I got into that was because forever before that, I wanted to be a surgeon. So I wanted to get into med school. I wanted to be a pediatric surgeon specifically. And so that's what I've always wanted to do. I studied through high school to get into medical schools. The way the system worked, there's a national exam, and I ranked enough where I got into private medical schools, which had higher tuition, and I ranked into dental schools, which is something I've never wanted to do. So my option was trying to figure out how I was going to pay tuition for these, private medical schools, or I could try to see something else where the path could lead me to some sort of research or medical related, work. So that's why I did my undergrad. So when I was doing my undergraduate degree, I knew about the basic biology. So I knew about immunology. I studied cell biology. I studied tissue engineering, which was, also fairly new. But I did not know about neuroscience as a field when I was in India, where I was and what I was exposed to, right? So I knew about the nervous system, I had learned in biology. So I knew the nervous system and all of the disorders related to the nervous system, but I did not know there was a field of neuroscience that I could study further on or do research in. And so when I came to do my master's, actually, the lab, my PI's lab, who ended up being my PhD advisor, his lab, he had a class for the master's students called Lab Principles. And in the Lab Principles class, if you've taken a class like that, you're looking at understanding how EEGs work, ECGs work, all of the kind of testing mechanisms of looking at, spirometry. Those kind of experiments is what we did. And so that really opened up a whole different kind of perspective for me, because I did not know something like this, which could connect engineering and basic biology or neurology really existed. So I got interested in his class. I did really well in his class. We had an term end project for that class, and the project we got selected for the university's conference symposium as one of the abstracts. And so that really then led me deeper into his work. And he was at the time doing a lot of spinal cord research, neuroregeneration research. And when I looked into that, I was really amazed by the details of the neurobiology of our nervous system, and it can be studied other than doing psychology or psychiatry, right? So the nervous system and treating the nervous system using basic biology knowledge and tools from tissue engineering. So it was kind of connecting the two for me in a way where I was like, okay, this is something that sounds interesting to me. So that's when I joined his lab as a master's student. So that really gave me a whole new introduced me to the whole new field of neuroscience and neurobiology, and also neurotechnology after that. So as I started working with him, I was looking at kind of helping on different projects in the lab. Then I looked at the bioelectronics side of the lab and what that really means. And you can really grow a nerve, you can really modulate a nerve, you can stimulate a nerve. Those are things that I learned when I joined that lab. So really, when I was doing my master's, that's when I got introduced to this concept of neurotechnology, studying neurobiology, but then applying engineering principles to modulate your neurobiology. That was very new for me. So that lab really, introduced me to this whole field, which is why I am here now talking about all these cool things, and really connecting my initial love of being in the medical field to what I'm doing now. So, I can connect kind of working in, back then I was really, as any student would, right? If someone doesn't get into med school, it's very disheartening. And so when I look back now, I really like the path I took, because it didn't lead me to med school, but it really led me to this kind of mix of biology and engineering, and that I can still help patients without being a doctor at the moment. So that one class in my master's degree, and when I joined the lab, that really introduced me to neuroscience and neuroregeneration, actually.
Yeah, that makes sense. And in my opinion, the field is lucky to have you. So I'm glad that you took that class and got introduced to all of these things.
Thank you.
And then another thing that really caught my interest during your background story there was how you mentioned that the spot where you work now is at the intersection of a lot of different fields, at the intersection of technology and engineering, as well as neuroscience, and probably lots of other different kinds of sciences are all feeding into it as well. And that's one of the things that really excites me about this field as well, how it's very interdisciplinary. It can benefit from innovation from lots of different directions. To dive into a little bit further detail on your background work, so you worked on an initiative a few years ago, and that included a publication exploring neuromodulation with a miniature wireless stimulator for small peripheral nerves. And I was just wondering if you could share a bit about the significance of those findings, as well as the future applicability of development such as the one that you and your team were working on.
So that paper, that was a paper where we were looking at this new electrode design, which was miniature in the sense that it was, close to, I think, 5 or 6 millimeters long in dimensions, and smaller than that in width. And the application we were really looking at in that paper is its neuromodulation effect on smaller pudendal pudendal nerves that could be stimulated to control urinary incontinence. If you think about your upper limbs or your legs, the nerves in your extremities are pretty large, and so even though they have the same kind of nerve and regeneration properties, they're easier to access. Now the nerves in the in the sacral plexus, or where the kind of the functioning of the ureter and the urethra lie, it's really like a plexus instead of a one whole nerve, and so it goes to dimensions of less than close to a millimeter in diameter, which is super small. And in order to, and bioelectronic medicine really is trying to go towards, a drug-free way to really solve a problem like urinary incontinence, which is prevalent in, hundreds and thousands of women, and, millions of patients if you count patients with spinal cord injuries, etc. So that was a problem, that is a problem still. And a lot of companies right now are targeting sacral neuromodulation to control for urinary incontinence. But that electrode, so the reason it was miniaturized was that it had a specific way to be implanted on a nerve that is small enough, and so that it wouldn't take up too much space where it was, because it was in a very awkward surgical location where there wasn't a lot of space to implant a big electrode that could do the work. And so that kind of design was so that it could be implanted well enough. It was quick implantation, and the efficacy of that electrode to be able to stimulate those kind of small nerves. And so I think in that work, what we did was first tested the efficacy of the electrode itself. So tested the electrodes on other devices that were more other nerves that were more accessible, and then the plan was to test it on those smaller nerve plexi, which could then very specifically stimulate those really, really small nerves in order to control the ureter and control bladder function, so that it could be controlled based on the patient's will, basically.
Yeah, absolutely. That's a super exciting development. I'm definitely happy to hear that that project was able to kind of take some of these paradigms and these stimulation methodologies that are being used on bigger nerves and use them to allow the patients to perhaps control some of these smaller nerves, like the ones you mentioned that were associated with the urinary incontinence. Another project that I wanted to ask you about was one that you were involved with that uncovered some direct vagus nerve to spleen communication a few years ago. That seems like a super exciting finding. And can you just talk a little bit further on the project that discovered that, and then also the applicability or just the significance of, like, now that that's been discovered, what can some of the future work be?
Yeah, absolutely. So that was one of the projects where what we were trying. So the cervical vagus nerve is the most targeted nerve in terms of neuromodulation for PTSD, or in general, when you hear vagus nerve, it is mostly the cervical location, so it's closer to your neck, and so the vagus nerve controls a lot of your intrinsic functions. And one of that was the hypothesis of that work was that it has a direct control over the spleen, and if we could really show that that it has this direct connection, then there was a way to specifically so since the vagus nerve up here is a whole branch of nerves, and as it runs down the body, it branches out and then creates all these connections. So there's two things we did. One was to show that we could really stimulate that and get signals from that. So for that, in order to answer that, we had to create a whole new type of device. So there's a electrode in there, which was it's called the sutrode. So it's the reason we call that is because it ties around like a suture would, but it is actually an electrode. So it was a graphene electrode that was like a suture that was tied around those nerves that went into the spleen. And these branches were like different. They had different functions, which was obviously, we found that later, but different number of electrodes were placed on those different branches there, and they were really small. So really no space to put a big device in there, which is why the function of those sutures or suture electrodes, sutrodes, was that to record the activity of those nerves as we stimulated the vagus. And there was so the different branches recorded differential activity as the main vagus nerve was stimulated, which means that they all innervate differently from the vagus and have different functions. And so we were, through histology and some retrograde tracing studies, we were able to show that they do occupy different areas within the vagus nerve itself, and that when those specific branches are stimulated, different areas of the larger nerve light up. And so it's all based on their neurobiology. So the way they are structured, the axon composition within those nerves also had a lot to do with how they were bundled up and went into the main branch of the vagus. And from there, our discussion was that there is a differential activity between these branches. And so now you can try to really if you wanted to control the spleen in some way, you could target these tiny branches, or within that vagus nerve to be able to control the spleen and its function. So it was a very basic science question. What do these nerves look morphometry wise? So what is the composition? What kind of axons it has based on myelination and axon size? But also, do they trace back to the vagus? Yes, they do. And when they're stimulated, when the vagus is stimulated, these nerves get stimulated in different patterns. So that's what, that uncovered a different mechanism of vagus and spleen kind of communication, which was, in the future, I think this was back when I was in grad school. Like I said, I did multiple things. And so this was part of that. And so right now what they're trying to do is, the first author from that paper, Dr. Gonzalez, she's working on kind of expanding this and really looking, she's a neurobiologist by training. And so she's really looking into the details of this, trying to uncover mechanistic, answers to this question, and also looking at how kind of we can tie up neuromodulation with that. Very exciting work.
Yeah, I agree. That is super exciting work. And I'm happy to hear that it's kind of continuing to move forward, and super excited to see what comes out next with regards to that work. And then I also noticed that you worked on a sutureless coaptation device for repairing peripheral nerve defects. And so I recall you mentioned when you were explaining the vagus to spleen connection, you mentioned that one of the tools that you were working with there was also sutureless. Were these tools related? And then also, how does the project differ when you're working on something like neuromodulation or recording versus repair of these peripheral nerves instead?
So actually, the vagus spleen project had a suture as an electrode. So imagine when surgeons are trying to suture a wound up, they're using these nylon or silk sutures trying to close up, and which are easy to put around, right? And so in the vagus spleen project, the electrodes themselves were like sutures. So they handled like sutures, and you could easily kind of loop them around the nerves, and they recorded like electrodes. So basically, they did the job of the electrodes while acting like sutures, which means that placing them around the nerve was very simple. In the project I'm working on right now, that's actually, I would say, the complete opposite, because we have no sutures in the device. So I'm working on this device called the nerve tape, and it's a sutureless coaptation device. So it is used to keep nerve ends together. So at the moment, the clinical gold standard is for highly trained microsurgeons to sew your nerve together. And for that, they have to use sutures that are thinner than a human hair, have to be really well trained to do these sutures in a way where the nerve ends meet. And peripheral nerves can regenerate, but obviously the regeneration rate and the regeneration end result really depends on how well the surgery is done. And studies have been performed where they looked at the quality of repairs by highly trained surgeons, and also by trainees like residents and fellows. And they saw that almost 40% of the times, regardless of whether they are highly trained or trainees, 40% of the repairs are not really a good quality repairs, which means that the nerves are not aligned properly, which means axons don't grow in the direction they're supposed to, which at the end means that there won't be enough functional recovery, right? So even with that, even with highly trained microsurgeons, there's suboptimal functional recovery after peripheral nerve injuries. I told you I work with a surgeon scientist. His name is Dr. Jonathan Isaacs, and he invented a device which is a fully sutureless device. So it has a flexible backing with metal microhooks, and these microhooks are really engineered to grab the outer epineurium of the nerve. So for someone who isn't aware of what the nerve biology looks like, you have this so the nerve is like a bundle of wires, and so the outside of the nerve is covered with a connective tissue called epineurium. Then you have your smaller fascicles, which means that fascicles are smaller bundles of axons, and these fascicles are kind of branched together. And these fascicles have an outside connective tissue layer, again, called perineurium. And then you have the axons, which are the single unit of the nerve, that are present within these fascicles, which are the conducting bodies of the nerve. And these axons will have the final layer around them, which is called the endoneurium. The goal is that when anything like sutures is put through a nerve, even the most experienced microsurgeon cannot control how deep you go into a nerve. And so most often you will find that sutures go through nerve, they create scar tissue. And so what he wanted to do was really solve this problem of, one, having to do such a technically challenging repair, being able to do it so that the repairs are always consistent, and to be able to do it in a way where the repair quality is always good, so that when the nerves do regenerate or the axons do regenerate across, they can regenerate to their full potential. Even with that, axon regeneration is not 100% complete. So even with all the ideal conditions, regeneration is still super slow, and there are other reasons for that. But one of the main things that really kickstarts it is a good connection of the two ends of the nerve and a good repair. And so the device, what it does is it has this flexible backing with metal microhooks embedded inside of the backing. And what that does is the metal microhooks kind of are on the level of between 300 and 500 microns, so super small, and they're angled. So what they do is they hold the outer epineurium of the two nerve ends together, which means that you don't need sutures to keep them together. You don't need you don't need to worry about the alignment, because you're not really, the device is aligning the nerves once you place them, and then you kind of wrap it around so it entubulates, which means that the nerve repair location, or coaptation, as it's called, is stable. And that provides, any sort of breakage or dehiscence, as it's called, which is very common in sutures as well. So if it's not a pretty strong suture, sometimes once the patients are closed up, there's a risk of the suture breaking off, which is why a lot of surgeons will, over tighten it, which is also one of the reasons why nerves don't regenerate well. And so this device does a good job of holding the nerve ends together without damaging the axons, because the microhooks stay only in the epineurium, and so it holds it together. So what we've done is through a lot of preclinical studies, we've shown that it's safe and it's efficacious. We've shown that it's comparable to sutures, and what kind of recovery we get after that. We've shown how consistent it is. It takes almost a fifth of the time to do the repairs, because it is, you don't need to be doing suture after suture after suture to be able to connect that. You just place it. And so it is easy. We've shown that it's easy. So right now it's super exciting for me as a scientist, because right now we're at a point where that product is going to be launched soon. We already have FDA approval. The product is going to be launched in a couple of months by the company who is our collaborator. So they are the ones who manufacture and are going to sell it and commercialize it. But we work together to do all of this testing and answer all of these, basic science questions about safety and efficacy. So it's super exciting, because I use a lot of, or all of, my PhD skills to do all of these validation studies, and now to be able to see that it's going to be soon put into humans is really an amazing experience that I've had in the past almost 4 years now.
Yeah, that sounds like an awesome project, and I agree, that's super exciting that the launch is coming up and it's getting kind of translated to humans, and everything's going smoothly with the FDA, and I'm super excited. Now that you've explained the details to me, I'm super excited about how it will, since you said this is a very specialized surgery when it's using sutures, I'm super excited how it could bring like the opportunity for more and more patients to get this surgery, and then also for the patients that do get the surgery, for their chances of it actually going well to be higher. So that's an awesome project. The more that you explain it, the more excited about it I get.
Yeah, that's the goal, actually. We are excited that once it's launched, we will really get clinical data that what we hope to see is that supports what we've done so far, and really shows that we've done a very thorough, scientifically rigorous studies. But at the end of the day, as you know, clinical is clinical, and as long as once we start getting our clinical data, that's when I will be more and more excited about it, because then we'll know more about how it works and how the future looks like soon. So I'm excited about it.
Oh yeah, absolutely. And I'm looking forward to keeping an eye on it and watching it go through these milestones going
Mhm.
forward. And so another question I wanted to ask you was, is there any other projects that you're working on that you'd like to highlight? And then also, what do you plan to work on going forward over the next few years with your research?
Right now we have, so our main focus is neuromuscular kind of repair. So what we're trying to do is right in very early stages, but what we're trying to do is look at, so once the nerve is injured, our first one of the first main focuses was having the nerve tape, where you're directly targeting how well the repair is actually done, right? Once the repair is done, then the nerve has to regenerate, get to the muscle. So there's two different kind of ways we address this. We're looking at different ways to, one, improve nerve surgery, especially in longer gaps. So right now, the application I told you about is when a nerve is cut or injured from, say, for example, a laceration or a gunshot wound, or not really gunshot wound, but a laceration or some sort of cut. That's where the nerve tape will be used at the moment. One of the other kind of clinical problems related to nerve injuries is when there's a larger nerve segment missing. So for example, in gunshot wounds, in motor vehicle accidents, you get injuries where there's a lot of debris in the surgical field. Then the surgeons can't tell how much of the nerve is damaged. There are other injuries that are causing the nerve to be resected out, and so in those cases what happens is there's a large segment missing from the nerve, and that's when surgeons have to look for other options where they will either use one of your own nerves. So the sutural nerve in the leg is the most commonly used nerve. They'll use that nerve as an autograft. Or there is a processed nerve allograft as an option. So there's a company that makes this, which has also shown a lot of great results in improving. So when there's no autograft available, or if it seems like it's a good a place for allograft, then that's what is used. So that's another question. So long gap injuries is still a problem, because what happens is even once you repair the nerves, if you use an autograft or allograft, depending on how well those options work, your muscle then regains function based on that. But if those don't work well enough, then your muscle is atrophying because it's not getting the conduction from the nerve. So it's not working. So it's atrophying as the nerve regenerates. So most often in these big injuries, by the time the nerve gets to the muscle has atrophied so much that now it no longer has any activity to be able to accept those nerve endings, make those connections successfully, and start working again, right? So it may work, but not to its full extent. But most often it has lost, the neuromuscular junctions at the end of the axons. Once the axons have died, those go away, right? So go away in the sense that they kind of collapse within themselves, which means they're not functional anymore. And so even if an axon comes there, say maybe 6 months after injury, that neuromuscular junction no longer is accepting axons to make that successful connection. And so one of the other things we're trying to do is trying to look at how we're going to solve this problem of muscle atrophy. And so some of the project, we're using different kind of ways to address this. So we're in a super early stage where we're trying to look at different modes where we could maybe give work on some drugs that can be injected in the muscle, or some sort of neuromodulation that can be done for the muscle to be able to keep up its mass and activity. And so, there's a lot of literature on that, but neither one solution has really worked well enough where it has, solved the problem clinically. And so that's what we're working on next. That's another kind of part of our research. And then the other thing is, since we worked on this nerve tape, we're looking at expanding the applications of the device, and so looking at other applications that where this device can be used. That's also, so we recently received a few grants where we're going to work on that. So that's called kind of also where our work is going, looking at other applications of the nerve tape itself.
Well, definitely lots of exciting research that you're working on now and going forward as well. And I did have one quick follow-up question with regards to the long gap injuries. That makes a lot of sense how those ones are super complex, and you're running into issues like these problems with atrophy and kind of working on how to solve that. I was wondering, when they're doing the grafting, are there any issues with like immune responses to like the grafting that's being put in as well?
So a lot of the surgeons, depending on how long that loss or gap is, will use autografts. So when they use autografts, which is your own nerve, then there's no issue with any rejection. But the allografts that are in the market right now, there's a proprietary technique to decellularize it. So there are no cellular components within the within the allograft. And so what happens is it has this bioluminal structure of a nerve, but does not have the cells inside it, which will then initiate the rejection. So there are no, so since it's decellularized, there are no host cells within there that would then in the person would elicit an immune response. So it's been developed in a very neat way where all of that has been kind of cleaned off, but the structure itself is the integral structure of the nerve itself is intact, which means that there are these biologically structured pathways for the axons to grow, which makes these allografts so successful. And recent studies have shown that allografts and autografts at certain lengths may be actually equivalent. So imagine if you have an injury and your doctor, if you were a scientist who knew about this and you had an injury, if the doctor asked you, should I take your own nerve and you lose some sensation in your foot, or should I use an allograft? And since most studies have shown that allografts are as good as autograft, you also would not want to lose that sensation in your foot, right? Most often when surgeons, there are no other better alternatives, which is why people are like, okay, fine, I'm fine with losing some sensation in the side of my foot if I can move my hand, enough to hold a bottle. When it's that kind of comparison, the perspective is that your function and your gross functions should be preserved as compared to some sensation in the side of your foot. But if there's an option to get the same kind of recovery without losing that, then obviously that's what you would want to do. So they have the process of cleaning and decellularizing has been done well enough where clinicians use this all the time, the allografts, without any rejection.
First off, thanks for explaining that to me. And then second off, that sounds like super useful technology to kind of have the biological properties that you want and need, but then decellularize and not have the types of properties that could cause issues. And then zooming out a little bit, I just wanted to ask about your thoughts regarding predictions for the field of neurotechnology over the next 20 years or so, let's say. And I guess I was wondering if you could speak a little bit on, first off, like the specific subset of the field that you do most of your work in, and then as well as in a zoomed out sense, and just kind of speak on the field at large.
So I think in terms of like zoomed into my specific field itself, I think there is, based on my personal experience, what I've seen since beginning my career as a student and now, is there's more dialogue between clinicians and scientists, and most often a good partnership, a working partnership. What that means is there's all of the sciences, most of the science is supported by clinical perspectives, which means that translating technologies has become easier than better because we work together from the beginning. And so I think in terms of my own field, I think surgeons are now open to going away from the traditional ways of repairing nerves and looking and kind of as basic science tries to answer more questions, looking at newer ways, using newer bioengineering technologies to repair nerves, right? That includes in the future using some sort of stem cells or some sort of spheroid. All of the technology is being developed. We as basic scientists have been studying these for a while, but now I just yesterday returned from a meeting that was it is a surgeon focused meeting in the sense that I had never been to it as a graduate student, but it had a lot of basic science sessions where we're kind of connecting those dots. We're bringing this lab bench technology to clinics, which which means that better collaboration, newer companies are formed to commercialize or to translate these technologies, which means that better options for people living with nerve injuries. And so that's what I think, that's where I think it's going. We still have, like, in the next 20 years, the way I see it is that we will, instead of having to use autografts or allografts, we would have something very specifically tailored to that patient. So something, cells from that patient that are tailored to regenerate the gap that that patient has. That's something I kind of foresee happening. So having growth factors, having a gap that is specific to that patient's injury, having cellular and growth factor components that are specific to that patient is how I see kind of this going in the next 20 years. And zooming out, last year certainly has been very interesting. A lot of focus on neurotechnology, brain-computer interfaces, but also bioelectronic medicine, right? For very, very basic clinical problems like chronic pain, migraines, urinary incontinence, things that were, that everyone knew was a problem before. Most of them were solved by medications. Most medications didn't work. Patients were frustrated. These were kind of conditions where patients had accepted that this is what these are the conditions they're going to have to live with. And now the way I see it in the next 20 years, one, there's a lot of patient education. There are a lot of great people, including, young scientists like you who are working towards this new dissemination of the correct information, right? So people know more about what's out there, what's going on. And so more patient education about, okay, this, using these opioid medications is not my only option. I could get an implant, which would be a small surgery, but I could be pain-free for a decade before I could get maybe an upgrade of that implant, right? And that's a perspective that is changing in patients as well, I think. And also in us, as technology gets better, we're, making smaller, more compact devices. We're looking at not only patient improvement, but also how the patient will accept the device. We're making it more convenient for the patients to use or not have to interfere at all or intervene. And that's something that I see happening too. But with that, what I think is the responsibility of the scientific community together is also to make sure that the science is rigorous and the data is what is in focus, instead of trying to go into a path where having publicity or being in the spotlight doesn't kind of deter you from what the actual goal is. And also, as it is with any other field, miscommunication and misinformation is a problem. And also, as we get more advanced with the brain-computer interfaces and more consumer-based devices, there's, again, this whole field of, data privacy, which is going to be very important. Just as we are, at the cusp of kind of, last year was a big year for AI as well. And now we are kind of realizing what we need to have in place to be able to have kind of the ethics in AI. And similar to that, there are a lot of great people who, there's a book on the neuroethics of the neurotechnology, basically, right? Neuroethics. So that is going to get more important as we develop these new technologies, so that we can kind of keep the balance on making sure we have data privacy or a consumer privacy, but also be able to help the people who need to be treated or need to have certain devices or certain products. But that's kind of my take on the field in general.
Yeah, I totally agree with you in terms of keeping scientific rigor and neuroethics at front of mind as we move into the future. But I'm also super excited about those other things that you mentioned, more personalization in the space of nerve repair, as well as the space of neurotechnology at large kind of being expanded for lots more indications. So definitely a bright future ahead for the space. In terms of the future ahead for the space, I was also wondering if you had any tips for young engineers or students or future clinicians or just for anybody in general who does want to get involved in, the nerve repair or the neurotechnology space?
Yes, I think it has been, it has been a field for years now, and it has been traditional in the most, in the sense that the treatments or the devices were traditional. But as more people started to focus on getting better, as we got better materials, better fabrication techniques, and better kind of software for signal acquisition and processing, we are getting better at making these devices better, right? So what I would suggest, so if I were, starting up right now as a student trying to get into this field, I would try to, follow the different companies, try to read about what they're doing, try to read about what they're up to, talk to more people. I mean, this is something that I learned a little bit later in life, but sometimes as a student, you don't think you have access to a lot of the people in a company, or scientists working at a company, or scientists working on a specific paper. But really, like, the reality is that it's a small world, and people interested in science always will find a way to get in touch with someone. What I mean by that is, now with everything, online communication has been made so much better now that you can easily find someone. If you find someone's paper, you can email them. If you find a company, you can add someone on LinkedIn, kind of get their perspective on things. So it's become it's easier to communicate with someone. So that's something I would suggest people doing, like, hey, I found this person, and that's what I do even now. I look at someone and say, oh, I like this person's career trajectory, and I will add them, I'll talk to them, I'll try to get some advice. And most often people are really nice enough to give you, like, if they don't have time, they'll give you like a brief answer and be like, oh, maybe look into this. And some people are really nice enough to give you a detail, like, oh, I'll be happy to jump on a call if you want to talk more, if you really need advice right now. So that's what I would suggest someone do. And in terms of like the basics on the scientist side of things, if you're a new student getting into research, you find a paper, you find it very interesting. I've had people, like, when I've published papers, I've had people email me about telling me about how interesting they found my paper, and had some questions about things that they probably didn't get from the paper, or weren't clear enough from the paper. And that has led to great discussions. And I, as an author, am always happy to get a paper email from someone who has read my work and is interested in it. And so if as a student you have any concerns about emailing someone, what are they going to think, will they even read my email, you'd be surprised how many times you will get a response back. So that's the one kind of advice I would give anyone, is to talk to that person, send a cold email or a cold DM now on LinkedIn, and you'll be surprised how many times you get responses. So I would have done that if LinkedIn was as good back then when I was a student, but I would not know that what I would not know what I know now back then. So I don't know.
Yeah, absolutely. You never know what a cold email or a cold LinkedIn message can lead to, and that's kind of how our discussion started. I was interested in your research, and I sent a cold LinkedIn message, and next thing you know, we're taping a podcast episode.
Yeah.
And I guess that kind of leads me into the next question. Can the listeners of the podcast contact you for further discussion after the episode? And if so, how can they reach out to you? And what sorts of topics would you want them to reach out to you about?
Yes, so they can reach, the easiest way to reach out to me is through LinkedIn. So you can easily find me. At least at the moment, there is only one Geetanjali Bendale on LinkedIn, and that's me. So until the next one comes along, you can message me and ask me anything about my research, about science in general. If you're getting into grad school, questions about grad school. I've had a lot of people message me on LinkedIn about, oh, I'm doing my master's in biomedical engineering. I want to get into neurotechnology. What should I do? I'd be happy to, answer any questions you may have. So, all the way from getting into grad school, what to do in grad school, to if you're at a point where you think, okay, I want to do what you're doing right now, I'd be happy to, answer any questions you may have.
Well, I think that was all the questions I had for you today. I'll be sure to link your LinkedIn profile in the show notes. I just wanted to say thanks again for coming on. It's been a super interesting discussion. I feel like I learned a ton from you, and I hope you have an amazing rest of your day and an amazing semester.
Thank you so much. Thank you for inviting me. This has been fun. I mean, it actually took me down memory lane, and I hadn't thought about a lot of the things you asked me today. So it was kind of refreshing to think about, me as a student and where I am now. So it was kind of fun to do. So thank you for kind of getting those memories fresh for me again.